Do a new one. No. I saw you. It's out for a minute. All right. Draw on the board. What did we do yesterday? What did we learn yesterday? We did talk some yesterday and caught up and welcomed and talked a little bit about what classes. Guys, why do I look so pale? Look at
Because
me. I look
it's
like a ghost.
January and we live in Ohio.
Guys, I just found
I like
something
look at
very
myself
bad.
for weeks.
I need to like add some massive like bronzer and lipstick or
I
something.
just found
man.
a very
Wow.
cool thing, and I just realized this. So if you use
What?
the text tool, and if you click,
Yes.
if you left click and drag, like it will put a text box, and then you could type on it, so it's easier. I just found that out.
That's awesome.
If you don't believe me, you guys can try it out.
No, we believe you, and we will try it out. We'll do both. Yeah. So we talked about mutations, and mutations are a what? This is right. This is a change. What's another M word that we used yesterday? Mutations are what? Thomas Schmidt. Yes, they are mistakes. They are mistakes in our DNA. How do they happen? Tell me some reasons they happen. They happen in our genes, to our genes. Perfect, Vinny. What else? How do we do them? They can be a copying error, right? Because we're like constantly making all these cells all over our body. So there's bound to be errors. What else? Yeah, because pieces of our DNA fall off. We are shedding DNA all over the place. Exposure to X-rays, right? Good. You're right. You're right, Christopher. What did we talk about yesterday with hazardous materials and going outside? Yeah, we're just talking, Ireland, about what we learned yesterday. Yeah, so when we go outside to the sun, we talked about how powerful the sun was last semester, that our plants harness those rays of the sun, right? They're little sun ray catchers. Our chloroplasts and our chlorophyll go out there, and I just picture them with little firefly jars, right? And they're running around and they're capturing them. They're putting them in the jars and they're using that to create energy for themselves. But they also, that energy from those rays can also do damage, right? Because when they hit our cells, and if we don't have sunscreen on or protection, they can cause mutations in our skin. Good. We talked about how smoking, right? Drugs and nicotine can cause mutations in our cells. Rob, and that cancer is one of those things. Is having a mutation always bad? Yes or no? I know. That's what I told him. Take care of your skin while you're young. Wear sunscreen. No, mutations aren't always bad. It's not always a bad thing, right? Sometimes you don't even know you have them and it's fine and it's whatever. And then other times it could be useful. And we won't know it's a mutation. We'll just think we're super awesome. And then there are times where it is bad. And we know it because it has negative repercussions on our body. Wait, did I start? Oh, I did start the recording. Sorry. Good. Ooh, and deletion. Somebody's putting the types. Good. So that is about as far as we got yesterday. Today we are going to look more specifically at what happens when a mutation occurs and a couple different forms of mutations. So let's go to, nope, not that one. Not that one. This one. All right. So there are 3 types of things that happen that are different types of mutations. 1, 2, and 3. Somebody else read these for me. Who wants to read? All right. Pip can read number 1. How about number 2? Ireland, number 2. And what about number 3? There's Aria for number 3.
As long as if there's someone new, it'll be me, then Ireland, and Aria. So let's begin. So, types of mutations. Gene point mutations, which involve one or a few nucleotides, adenine, thymine, guanine, and cytosine. One of these is substitution, substituting one base for another, sometimes called silent because it doesn't always change the protein being made. You know what they say, silent but deadly? Well, that's kind of the thing. Anyways, take it away, Ireland. And thank you.
Um, insertion, one base is added to the chain.
Perfect. And Miss Aria, number 3.
Oh, you're muted.
There it goes. Okay, there it goes. Okay, deletion. One base is deleted from the chain.
Perfect. All right. So I want you to think about how this is affected with. Hey, Mila, I think there's a necklace on the laundry room counter. Can you go grab me one? I'm trying to think of a good visual for you because this. So the substitution is this, like when one of our base pairs just gets exchanged, right? It's just like an exchange. If you got something for Christmas that you didn't like, right? So like you got a pair of sweatpants and you're like, these are really cool, but I hate this color. So you take them back to the store and you just swap out the pair, right? This is kind of what happens, minus you don't get to pick your nucleotides, right? So that it just is an exchange. Substitution is an exchange of nucleotides, and it can change what is being made, and sometimes it can't. It could be like maybe you substituted, like in a recipe, salted butter and margarine, right? Like they're almost the same. They still like helped you get what you need for the recipe. It still made your cookie. There might have been a slight difference in taste, but it still made the cookie because you still had some type of butter. All right. That's substitution. Okay. I love my Alexa for that because, like, when I'm cooking and I run out of something, I'll be like, Alexa, what can I use instead of baking powder? And it'll tell me something that I can use instead of my baking powder because I'm out of baking powder because that's how it works, you know? And so a substitution is just like that. I want you to think about that. Asking Alexa for a substitution. Now, sometimes it can be vital and it doesn't work out, right? And then your cookies fail and it's sad. Insertion. All right. So if you look at this necklace, there is a pattern or there's a sequence of beads on this. Okay. Insertion is when none of these beads are taken out. Okay, none of them are taking out. So it's not going to mess up the length that this already is. In insertion, you legitimately just add it. Now, if we needed 10 and we added one more, then what is 10 plus 1? 11. All right, and if this is a pattern and we put one right in the middle, what is that going to do to our pattern? What's it going to do? I'm not doing a very good job of this. No. But if we plopped one right in the middle, yeah, it's going to change the pattern. Does anyone know what it's called? What is that called when one thing comes in and disrupts the pattern? Breaks the chain. I kind of like to think of it like the domino effect. Have you ever heard of the domino effect? Because the base pairs match up. If we squeeze one in the middle, then it throws off all the base pairs after that. Does that make sense? It's going to not just affect that one spot. In substitution, it affects that one spot. But when sorry, Mila's watching. She's in science with us today. My stage hand. All right. But when you put one in, when you insert one, a whole new one in right here, you're not getting rid of the A or the T, but you're putting one in, it shifts all of those down the line. Does that make sense? Give me a green check or red X. Because that is what is the big deal about insertion and deletion. When you take one out and they all shift together, or you add one in and they all spread apart, from the points where that insertion or deletion is made, it has the domino effect. And now you've got all of these problems. You don't just have one base pair problem, you've got a whole nasty chain of mutations. Okay? Most of you are following, I think. So let's do this because this makes sense to me. I like to I have lessons mapped up when we come here, but then when I'm working through these with you, I'm like, well, this would be a good idea. All right, so let's do this because you guys did this last semester. If you weren't here last semester and this is confusing, this will be a good review. All right, in our DNA, we have nucleotide bases. All right, we're going to do DNA. That was not a very good shape. We want it to be the exact same size. All right. I want to put a sequence down, and then I want you all to fill in the opposite pairs. Does that make sense? I wish I could do this quickly and more pretty, but it's not going to be. We're going to see what happens. We're just throwing these in. This is our DNA. This is one side of the ladder, right? We are going to create a thing. So this is let me put my cap locks on. Adenine, thymine, cytosine. Let's do, oh, now my text boxes are getting too big. Come on. Love me, Neuro. Love me. Let's do another C. Let's do what do you think? Do another A. And lastly, we'll do another G. All right. So this is one side of our DNA. Pretend those are all in line, but my text boxes were just being weird and not nice. And so that's just how it looks. Not linear. All right. This is one side. We're going to now make the other side, and I want you to, let's match up. What does A pair with in DNA? A pairs with what? Everybody tell me in chat. A always pairs with T. Perfect. So we're going to put in T. Now, instead of doing what happens, we're going to pretend that this is the shape that ruins it all. All right. This nucleotide, I want to actually get rid of that, is going to be the one that pops in. We're going to do a situation where this is insertion. Okay. And we're going to make it red because it's like, stop, this is the problem. All right, this is going to be the one that pops in and messes up the whole chain. So let's look and pair now. What goes with T? Let's match these together. Somebody put what would normally pair with T in this box. Will someone put an A here? Yep, good. What pairs with C? We're going to get rid of this one right here because this is going to be our mutation. We're going to pretend this is A that goes across from A. Let's pretend, Island, can you change that to A in the red box? Can you change that to a C? All right. Hold on. This is G. Good. Can somebody put what pairs with T? Right here. What pairs with T? Put an A. Good. What pairs with C? Put it in this box that I'm making. C and G. Good. Adenine pairs with what? Goes right here. T. Good. You guys are rock stars. All right. So this is our original strand. This is the normal strand. And this was all beautiful. This was all a beautiful situation until the problem child. Everybody point at the red nucleotide. We inserted, well, I mean, we don't necessarily ignore this one. It goes over here and it doesn't get a match. So G would pair with C. So see, what happens is when we inserted that random nucleotide, the pain point where it actually started was right here. But instead of just affecting this one spot, it affected all of these base pairs. This is now all kind of ruined DNA. Right? Because look, this C doesn't even have a pair. And if it had a pair, we'd be lucky if it didn't just take it off, right? Good. Right, Pip, you're right. So the combination that would normally have been correct is now out of sync. So what happens is our substitution, when only one base pair gets messed up, that is our best case scenario mutation. When you have insertion or deletion, you've got like just kind of major problems from that point forward. What they call this, guys, is it's not super important, but it's good to know if you can remember, is called a frame shift mutation. Right? If you see that, that makes sense, right? A frame or a whole set of DNA gets shifted and moved because of one insertion or deletion. All right. Yes. And those often end badly, right? They don't usually end without there being a consequence genetically, right? So those are our three types. You're going to want to know those. Substitution, right? When we're just doing an exchange, insertion, and deletion. And this kind of is just, there's our frame shift mutation, right? We talked about substitution being sometimes the silent mutation that you don't know you have. So let's look at a couple examples of mutations. One of those is called sickle cell anemia. Has anyone heard of that? Give me a green check if you've heard of sickle cell.
Yeah, I watched a TED Ed video on it, and I'll probably send you the link. And
Yes.
speaking of which, maybe this one too, because I know y'all love the Amoeba sisters, and I've seen them like years ago. So
Yes. So sickle cell anemia is a disease that we can have, and it happens because of a gene point mutation. All right. Again, we talked about different people being more susceptible than others to this. All right. But what happens in sickle cell is, look right here. Let's look at this again. This is what the normal DNA reads. And this is what happens in sickle cell anemia. All right. Instead of that T, there is a switch to an A. And then when we switch from our mRNA to our amino acid, look what happens. Because see, remember our DNA, a copy of our DNA is made from our RNA. That's our recipe card that we take out of the nucleus into the cell. And we take this mRNA copy to our ribosomes to make our proteins. But look what happens when that one substitution is made, right? The T is no longer a T, it's an A, which our mRNA codes for a U. And we take it to the ribosome and it codes for an entirely new, you see valine here versus this glutamic acid. And it's a whole different, this is the cell that it codes for. This is what a red blood cell typically looks like. And this is what is made in sickle cell. All right, that's what they actually call it. It's the sickle cell. Kind of reminds me of a sick cell, right? Let's also look at cystic fibrosis. So sickle cell is an example of what kind of mutation? Insertion, deletion, substitution. Tell me in chat. It's supposed to be a T, but it's an A. It's just exchange for another nucleotide. Yes. Substitution. All right. So sickle cell is a point mutation, a gene point mutation, and with one is substitute for the other. So let's look at cystic fibrosis. All right. Cystic fibrosis is caused by a mutation on our 7th chromosome. All right, our 7th chromosome. And cystic fibrosis is an example of deletion. Deletion. All right, so I want you to remember that cystic fibrosis is associated with deletion. All right. It is the most common mutation that causes cystic fibrosis. There are other things that happen that can cause that, okay? But there is deletion. So this is normal, TTT, the codes for this. And then this is what it looks like in cystic fibrosis. So we're good here. Look, those match up. These match up. These match up. And then, oh no. Right? Here we go. And we're lucky here that it looks like it's just this triad. See how that these still, oh no, wait. Yes. Well, I guess I take that back. It's not, because look, see, these all are the same here then afterwards, but this one is missing. All right, so the deletion of that amino acid, of that whole triad, is what causes cystic fibrosis. All right, it changes the shape of our amino acid, and that is what is so dangerous. All right, so cystic fibrosis is an example of deletion. Whoa, that was a crazy, that was supposed to be an underline. Sickle cell is going to be our substitution example. And then let's look at one more. Are we at one more? We're going to look at some chromosomal abnormalities. All right. So somatic cells, where are those in your body? Give me an example of a somatic cell. We talked here about this yesterday. What's a somatic cell? Tell me, where are they? You got this. Yeah, your organs. Can be your skin, it could be your eyeball, it could be your liver, it could be your stomach, it could be anything. All right, other than your sex cells. So you've got your somatic cells and your germ cells. All right, the somatic cells are all of the cells in your body. All right, and when there is a mutation that occurs in your skin cell or your liver cell or your eyeball cell or your brain cell, all of that, those mutations, they only affect what? You. Because they happen in cells that are only going to be reproduced in your body. However, when you have a mutation that happens in a germ cell, all right, a germ cell is a sex cell, right? So those are your egg cells, females, and sperm cells, males. When there are mutations that occur in our reproductive cells, then you can pass that on. Right? That's like how you inherit necessarily a mutation. Does that make sense? Because we talked about that yesterday. Can you inherit something that's a mutation? Well, typically not, because out of all of the cells in your body that could mutate, 99% of them are not going to be passed on, right? You only have one type of cell in your body out of all the bazillion of cells that you have that's going to be passed on, and that is going to be your chromosomal cells. All right. Awesome. So let's move to our quick check. I'm going to remove these. Whoopsie. Dun, dun, dun. All right. So let's do this. Oh, wait. Wrong pointer. I'm like failing at neuro today. All right. So let's have someone read the first question and I will put up our polling. And that's how we'll answer that. All right, Pip, go ahead and read the first one. And Ireland, go ahead and read the second.
All right. A geneticist noticed that a particular mutation had no effect on the polypeptide coded by a gene. This mutation probably involved either one of the following: A, substitution of one nucleotide; B, B, deletion of one nucleotide; C, insertion of one nucleotide; or D, alteration of one nucleotide. Thank you.
Good. This is kind of tricky. I kind of don't like this question. So for simplicity's sake, I'm going to just do this. I'm going to get rid of this one. Does that help you? You can change your answer, hopefully, maybe. Let's pick from the three types that we've talked about, because the alteration as substitution kind of reminds me of the same thing. I think that's kind of silly, right? But yes, the answer is A. It's a substitution. When we exchange that nucleotide out, that is the least likely to have a negative effect on us, right? Sweet. All right. Ireland, will you read number 2 for us?
A person with cystic fibrosis is missing blank. A, the contractile protein used in muscle movement. B, the gene that codes for an important cell membrane protein. C, an important mucus. Or D, an important protein easily obtained by food.
All right, so let's go back up here. What are they missing? The mutant form prevents chloride transport, causing mucus buildup. So what do you think? Going back down. I can't find it all of a sudden. Here it is. Which one do you think it is?
When I looked at the question, for a second I thought there were 2 A choices, but boy was I wrong.
Yes. And I don't even love these questions, to be honest, because they're not. I want you to know what the gene is. I care that you know what cystic fibrosis is, but I mostly care that you know that cystic fibrosis is caused by what. You guys are correct. By which one? Substitution, insertion, or deletion? Good. Deletion. All right, we have 5 minutes. I just want to introduce this for a minute because we're behind schedule, because I just miss you guys and I want to talk to you. And so that's what we did. All right, so let's just go here for a minute, right? We all know what this is. This is a cell, right? We are in biology. We're looking at cell. It is the smallest living thing. Yesterday, we talked about my house, right? I have these walls on my house because they keep me safe, right? That's why I live in a house with walls. They keep me warm. They protect me from the wind, the rain, the elements, the animals. It keeps them out of my house. And that's what the nucleus does for our DNA, right? It keeps all the things out. It protects the most important thing that makes us, which is our DNA. Because if a cell doesn't have its DNA, can it live? Can a cell live? A cell cannot live if it doesn't have its DNA. All right, inside the nucleus, our DNA is condensed into chromosomes. And if you pulled those chromosomes apart, right, it would be a double helix. A double helix is the same thing as like if you think about a ladder, right? We all know what a ladder is, twisted. That's what a double helix is. All right. And then inside of that, each rung of those ladders has a gene on it. All right. That gene is a section of DNA made up of 2 nucleotides, and it codes for something that makes us. What is something that about you that DNA codes for? Tell me. Hair color. That's an example. Type in chat something that DNA codes for in you. Eye color, hair color. How about height? Sometimes it has to blood type. Some of your weight tendencies are controlled by your DNA. The tone of your voice. Yes, our hitchhiker's thumb.
The tone of my voice. Who
Yes,
thinks
whether
I
you
sound
can
like a guy?
roll your tongue. All kinds of things, right? Our DNA also codes, we talked about proteins. That was like where we ended before Christmas, is that we go to our DNA. Our DNA is like our recipe book. We get sections of that DNA, sections of those genes to tell our body how to make more blood cells, make more skin cells, hair cells, muscle cells, brain cells, nerve cells. Our body is constantly sending little messengers to our nucleus to get copies of recipes for how to make things, and then our body is making them. A lot of these jobs are controlled by proteins. Okay? Proteins. Proteins are in charge of all of these things on the slide. So this would be a good slide to take a quick screenshot of. All right. Proteins have a bazillion jobs that we need them for. Now, sometimes we need those jobs to happen all the time. And sometimes those jobs, we do them, and then we need our body to stop doing them, right? So what happens is our body actually has in our DNA I want you to think about a wall full of light switches. I want you to look at a wall in your house and right now in your room, wherever you're at, and pretend there are just a bazillion light switches on that wall. All right. Our bodies turn off and on all these light switches all the time to tell our body when to start making things and when to stop making things. All right. We're not going to go over this right now. We did talk about this a tad yesterday about how our body gets those copies, right? There's our DNA. I need this section of my DNA to make whatever my body needs. So we do that by making a copy of mRNA. The copy of the DNA comes out and goes to this ribosome, and then it makes that protein to do whatever we need our body to do. Now, this is sometimes when things go badly, when those switches don't turn off or don't turn on. Can someone read this slide for me? Go for it, Ireland.
While it is true that each of your cells has all of your DNA genes, not all of your genes are expressed at the same time. You would not want the genes in your stomach cells that create an acidic environment for digestion expressed in your... Ouch! Acid in your eyeballs?
I know, right? So you've got the same type of cells in your stomach as you do in your eyeballs. So technically your eyeballs could produce your stomach acid if we told them to. However, that would really hurt and we don't want that, right? So our cells, depending on where they end up, right? They're made wherever they end up, whatever their job ends up being. Our eye cells say, stop remembering how to make that stomach acid. We don't need that. We don't need that, right? We don't want you to do that now because your job is not a stomach job. Your job is an eyeball job. So what our body does is it does have to turn off and turn on these different traits at different times depending on where it's at. So, this quick check really quickly. In the body, why do muscle cells and skin cells behave differently? Well, they behave differently because let me see. They have different genes. No, they all have the same ones. The genes are being expressed differently. The same genes are being expressed in both cells. There are no similarities. All right, the genes are being expressed in both cells. Okay, in different ways. Which genes are present in liver cells? We're just straight out of time. Let's go over this tomorrow instead of just trying to. Yes, this is today's quiz. So take today's quiz. Okay. And then we will go over this tomorrow and you can fix it if you need to. Does that make sense? Sorry, I'm just rushing and I'm just going to rush it and then it's not going to even make sense. And then we're going to be doing it again tomorrow anyway. All right, so take this quiz. You can take it unlimited amounts. So take it today. If you're like, oh man, I just blew that. Don't worry, we'll come back and we'll take it. And I'll even let you take it tomorrow during class. Wait, tomorrow is... I'll do this during the help session tomorrow. Okay. Sweet deal. I'll also send out an email. And I'll do a little video clip explaining these. How about that? I'll make sure it says more, Aria. I think its default is to 2. But I'll make sure it's unlimited. Because that's the way we roll around here. I want to make sure you know what you're doing versus just guess and go. I'd rather you learn it and instead of being like, what happened? Why do I not know? I'm going to do that right now before I forget. And then that doesn't happen because that's not cool. Unlimited is officially changed. So you should have that now. All right, you all. I have another appointment. Perfect, Pip. I will check. So send me any questions you have because I have another appointment to head out to. Have a wonderful day. Thanks for being here, guys.